Hello everyone and welcome to our channel, Mootechie. A special welcome to all the dairy technologists and students with us today. We're going to take a detailed look at a critical process in our field, industrial sterilization. So what exactly is sterilization? Sterilization is a technique used to make anything free from microorganisms, either by removing them or by killing them. In fermentation, the main aim of sterilization is to remove or kill all organisms in the medium. If we don't, contaminants can seriously deteriorate the entire process. Contaminants can enter a fermenter from many points. This can happen through improper sterilization of the media, partial sterilization of the air, the cooling water system, through various inlets and outlets, due to a faulty inoculum procedure, because of the process used to pre-treat crude ingredients, or from a leak in the fermenter's design. Why is sterilization so necessary? If a foreign microorganism invades the fermentation, the desired product may not be produced. It is absolutely essential to sterilize the medium and all materials so that only the specific organisms we inoculate will grow, giving us a high yield of our fermentation product. Therefore, sterilization plays a vital role. The consequences of contamination can be severe. The medium would have to support the growth of both our production organism and the contaminant, leading to a loss of productivity. In a continuous fermentation, the contaminant might outgrow and displace our production organism. The foreign organism could contaminate the final product itself, which is a major issue in products like single-cell protein. The contaminant might produce compounds that make extracting our final product very difficult. It could even degrade the desired product, which is common in antibiotic fermentations, where bacteria can produce enzymes to break down the antibiotic. Furthermore, contamination of a bacterial fermentation with a phage could cause the culture to lyse or break apart. We can avoid contamination by taking several key precautions. These include using a pure inoculum to start, sterilizing the medium, sterilizing the fermenter vessel itself, sterilizing every item used during the process, and maintaining aseptic conditions throughout the fermentation. The principle behind sterilization is to remove microorganisms that would otherwise cause problems. There are two main methods: destruction of microorganisms and removal of microorganisms. Sterilization can be carried out by several methods, such as filtration, radiations, ultrasonic treatment, heat treatment, and chemical treatment. The extent of the procedure we adopt depends on the likelihood of contamination and how serious its consequences would be. The microbes we need to worry about include bacteria, fungi, protozoa, spores, and phages. Destruction of microbes is often achieved by physical or chemical means. The most common physical agent in industry is moist heat. Removal of microorganisms is typically done by filtration, which depends on the size of the microbe and the effectiveness of the filter. How strict we need to be with sterilization depends on the type of fermentation process, how long it runs, and the nature of potential contaminants. Some fermentations are considered protected. This means the medium can only be used by a very limited range of microorganisms, or the growing organism creates selective conditions like a drop in pH. Beer brewing is an example. Hop resins inhibit many microbes and the yeast lowers the pH. So worts are boiled but not fully sterilized, and fermenters are cleaned with disinfectant but not necessarily made sterile. The precautions for the inoculum are also less strict compared to, say, an antibiotic fermentation. However, most fermentations are not protected and would suffer if contaminated. So what articles need to be sterilized? We need to sterilize the media, the air, the fermenter itself, and any feeds or additives. Different articles are sterilized by different methods. Heat-labile media, like serum or animal tissue, is sterilized by filtration. Heat-stable media is sterilized by steam under pressure. Air is mainly sterilized by filtration. Media sterilization can be done in two ways: as a batch process or as a continuous process. The design of the fermenter is crucial for proper sterilization. It must be designed to be sterilized with steam under pressure. The medium can be sterilized inside the vessel or in a separate vessel and then added aseptically. If done inside, the fermenter should be heated by steam in its coils or jacket first to prevent excess condensate. All entry and exit points are potential contamination sources and must be sterilized with steam, except the air outlet which lets steam escape. Pipeline construction should be simple, without slopes towards drains, so steam contacts every part. Steam should be trapped at every drainage point. Experts have long insisted on removing fine crevices where media and microbes can accumulate. For pilot-scale fermenters using animal cells, surfaces must be free from gaps larger than 0.05 nanometers. Welded joints are best for long-term aseptic conditions. The fermenter can be sterilized separately for aseptic operations. Sterile medium from a separate cooker can be added to a sterile, empty fermenter. Alternatively, the medium and fermenter can be sterilized together in one process. Not all fermentations are fully aseptic, but contamination must be minimized through methods like boiling or pasteurization. Otherwise, fast-growing contaminants will consume the nutrients. For small-scale fermenters, both the medium and vessel are sterilized together in an autoclave using steam at 121 degrees Celsius for 15 minutes. It's critical to avoid negative pressure inside afterward, which could draw in unsterile air. If the medium is sterilized separately, the fermenter is sterilized by passing steam through it for about 20 minutes at 15 psi. Afterwards, sterile air must be sparged in to maintain positive pressure. Now let's talk about filter sterilization of fermentation media. A fermentation medium contains many ingredients like carbon sources, nitrogen sources, minerals, and anti-foaming agents, each with different heat sensitivities. For heat-labile components, sterilization by heat isn't advisable, and filtration is the wise choice. This is especially true for animal cell culture media, which contain heat-labile proteins. Filter sterilization depends on the content, size, type, and moisture of the medium. There are different types of filters. Filters with a pore size smaller than the particle size are called absolute or fixed pore filters. If undamaged, they can remove 100% of microorganisms. Filters with a pore size larger than the particle size are depth or non-fixed pore filters, made from materials like felts, asbestos pads, or fiberglass. The terms can be misleading, as absolute filters also have depth where filtration occurs. Suspended solids are separated from a fluid during filtration by several mechanisms. Inertial impaction happens when particles, due to their momentum, travel in a straight line and impact on the filter fibers. This is more significant for gases. Diffusion affects very small particles subject to Brownian motion, causing them to deviate and hit fibers. This is also more important in gases. Electrostatic attraction occurs when charged particles are attracted to opposite charges on the filter. Interception happens when particles larger than the pores are directly trapped, but smaller particles can also be caught if they bridge a pore or if a pore becomes partially blocked. This is equally important for liquids and gases. For media sterilization, fixed pore or absolute filtration is the better system. An ideal system for animal cell culture must produce medium free of fungal, bacterial, and mycoplasma contamination, with minimal protein adsorption and free of viruses and endotoxins. Manufacturers supply systems with a series of membrane cartridges in stainless steel modules. A single filter can't do everything, so a sequence is used. For example, a system might use a 5-micron pre-filter to remove large contaminants, a 0.5-micron filter for bulk microbial removal, a 0.1-micron filter for further cleaning, and another 0.1-micron double-layered filter to remove mycoplasmas and ensure sterility. A final 0.04-micron filter would be added for virus removal. Similar systems are used downstream to remove cells and debris, using a 1-micron pre-filter followed by a 0.2-micron filter for absolute cell removal. A filter cartridge has a pleated membrane supported by a structure. They come as cartridges for stainless steel housings or as pre-sterilized plastic capsules. Moving on, we explore the methods for medium sterilization. Media can be sterilized by filtration, radiation, ultrasonic treatment, chemical treatment, or heat. Heat or steam is the most commonly used method. The success of heat sterilization depends on the number and types of microorganisms present, the composition of the medium, and its pH and particle size. Filtration is the exception for heat-labile components. Sterilization by steam is a first-order chemical reaction. The rate of organism death is given by the equation, where N is the number of viable organisms, T is time, and K is the specific death rate. Integrating this gives, taking natural logarithms, we get a plot of the natural text log of n underscore t over n underscore 0 against time gives a straight line with a slope of minus. This model predicts that sterile conditions are achieved only after infinite time, and that after a certain time, the number of cells will be less than 1. Continuous sterilization has several advantages over batch sterilization. It offers better protection of the medium's nutrient value, is easier to scale up, allows for easier automatic control, reduces the need for steam flow, shortens the sterilization cycle time, and can reduce fermenter corrosion. However, batch sterilization also has advantages, including lower equipment costs, less chance of contamination, easier manual control, and being more suitable for media with high solid content. Now let's look at the design of batch sterilization processes. The main aim is to achieve the necessary level of sterility with the least possible damage to the nutrient value of the medium. It's important to note that the continuous sterilization process is generally better than batch sterilization at avoiding nutrient damage. In batch sterilization, the maximum temperature used is typically 121 degrees Celsius. Therefore, the method is designed to expose the medium to this high temperature for only a short period. This high-temperature short-time approach is achieved by carefully considering the heating and cooling phases of the sterilization cycle. Researchers Daendorfer and Humphrey in 1959 offered a method to evaluate the important role that these heating and cooling periods play. Key points for batch sterilization. Moving on, we will see the key points to consider for a batch sterilization process. First, we must know how much the temperature of the fermentation medium increases during the heating phase and decreases during the cooling phase. Second, we need to know the initial number of microorganisms present in the medium. And third, we must understand the thermal death rate of the specific organism we are targeting. The required del factor is designed by knowing the initial number of organisms and the acceptable risk of contamination. A commonly accepted threat level is 1 contamination in 1000 batches. This means that after the sterilization time T, the number of living organisms should be 0.001. For example, if an unsterile broth contains 10 to the power of 11 cells, which is 100 billion cells, then the del factor required for that situation is 32.2. However, it's crucial to remember that the killing of cells happens not only during the holding period at 121 degrees Celsius, but also during the heating and the cooling periods of the process. Therefore, the overall del factor is the sum of the del factors from the heating period, the holding period, and the cooling period. By knowing the temperature profile and the time it takes to heat up and cool down, we can determine the del factor contributed by these periods. From there, we can estimate the specific holding time needed at the target temperature to achieve the overall required del factor. Methods for batch sterilization. Next, we explore the methods for batch sterilization. The sterilization of the fermentation medium can be achieved either directly inside the fermentation vessel or in a separate vessel called a mash cooker. Richards in 1966 evaluated the relative merits of in situ sterilization versus using a separate vessel. Advantages of a separate medium sterilization vessel. There are several advantages to using a separate medium sterilization vessel. 1. The medium can be sterilized in a more concentrated form and then diluted in the fermenter with sterile water before inoculation. This allows for the construction of smaller, more economical cookers. 2. One cooker can serve several fermenters, and the medium can be sterilized while the fermenters are being cleaned and prepared for the next run, saving significant time between fermentations. 3. In some cases, the medium is at its most viscous during sterilization, and the power needed for agitation is high. A central cooker with a powerful motor can provide sterile medium for multiple fermenters, reducing overall energy needs. 4. Finally, this method spares the main fermenter from potential corrosion caused by the medium at high temperatures. Disadvantages of a separate sterilization vessel. Coming up, we find there are also disadvantages to a separate sterilization vessel. 1. The cost of constructing a batch medium sterilizer is often similar to the cost of a fermenter itself. 2. If one cooker serves many fermenters, it requires complex pipe work to transport the sterile medium, which increases the inherent danger of contamination. 3. A mechanical failure in a central cooker that supplies multiple fermenters would make all of those fermenters temporarily redundant. Providing backup contingency equipment can be prohibitively expensive. Design of the continuous sterilization process. Coming up, we discuss the design of the continuous sterilization process. The planning for a continuous system follows principles similar to batch systems. In continuous sterilization, the medium is heated to the sterilization temperature of 121 degrees Celsius, held at that temperature for a specific period, and then cooled down to the fermentation temperature. The heating is done in a continuous heat exchanger, and the temperature is maintained in a long, winding holding coil. The length of the holding time is determined by the length of this coil and the speed of the medium flowing through it. After the holding time, the medium is cooled using two heat exchangers in sequence. The first heat exchanger uses the incoming cold, unsterile medium as the cooling source. The second heat exchanger uses cooling water. Advantages of continuous sterilization. The continuous process allows for the use of higher temperatures, which reduces the required holding time and minimizes nutrient loss. The necessary del factor is achieved by selecting the proper temperature and holding time combination, which further decreases nutrient damage. Additionally, a continuous process only heats and cools a small amount of medium at any given moment, which is much more efficient compared to heating and cooling an entire batch at once. There are two primary types of continuous sterilizers: the indirect heat exchanger, and the direct heat exchanger, also known as a steam injector. The indirect heat exchanger double spiral type. Let us go further and examine the indirect heat exchanger, specifically the double spiral type. The most appropriate indirect heat exchanger is the double spiral type. It consists of two sheets of high-grade stainless steel molded around a central axis to form a double spiral. Steam and the medium pass through the two separate channels in opposite directions, which allows the medium to reach the sterilization temperature. This same sterilizer can also be used to cool the medium after its holding time. The incoming cold unsterile medium is partially heated by acting as the cooling agent for the hot sterile medium leaving the holding coil. A schematic diagram shows a spiral heat exchanger. Another diagram illustrates the system flow, including a cooler, a cleaning cycle with cooling water, and the path of sterile medium to the fermenter. It also shows a heat maintenance unit and a heater for recovery of residual heat, all connected to a medium reservoir. An image of an industrial-scale spiral heat exchanger is also provided. Advantages of the spiral heat exchanger. The major advantages of the spiral heat exchanger are now outlined. Point A. There is less chance of contamination between the medium and the cooling liquid or steam, because they flow separately in compartments formed by stainless steel plates with gasket seals. Point B. The exchanger is kept clean by the scouring action of the steam, the cooling liquid, and the continuous movement of the media. This reduces the chances of sedimentation, fouling, and burning on. The alternate plates in the heat exchanger allow two different fluids to circulate in opposite directions. However, the two plates are divided by a gasket, and a problem with this gasket could lead to cross-contamination between the streams. This sterilizer is best for completely soluble media, as any suspended solids could potentially block the narrow passages between the plates. The capacity of this sterilizer can be increased by simply adding extra plates to the assembly. Advantages and disadvantages of the indirect heat exchanger. Following this, we notice the advantages of the indirect heat exchanger. Advantages include immediate heating up times, the ability to sterilize media containing solids, less investment cost, easy maintenance and cleaning, and high efficiency in using steam. Disadvantages are that heating may cause foaming, and since steam is in direct contact with the medium must be concentrated enough to account for condensate dilution. The steam must also be clean and free from any anti-corrosion additives. The direct heat exchanger, steam injector. Now let's look at the direct heat exchanger. In a direct heat exchanger, the medium is heated by injecting steam directly into it. Cooling is achieved by the sudden expansion of the medium into a vacuum chamber. This cooling happens almost instantaneously. A flow diagram depicts a typical continuous injector flash cooler sterilizer. It shows cooling water coming in, a cooling heat exchanger, a preheat heat exchanger, and steam and medium inlets. The sterile medium exits after passing through a Venturi valve, a holding coil, an expansion valve, and an expansion chamber. Steam is sent out to a vacuum system. Another flow diagram shows a typical continuous sterilization system using spiral heat exchangers. It includes inputs for water and detergent, steam lines, and the path of the sterilized product to the fermenter. The system features a holding tube, control units, cooling water, a flow control valve, a balance tank, a spiral heater, a spiral heat interchange unit, and a spiral cooler. The raw product enters the system from a reservoir. The process is initiated by passing hot water through the entire system to sterilize the plant before the medium is introduced. Steam is then used to sterilize all the pipework and the fermenter. Heat is conserved efficiently by using the incoming cold media to pre-cool the outgoing sterile medium, which in turn preheats the incoming stream before it reaches the main sterilizer. Advantages and disadvantages of the continuous steam injector. The advantages of the continuous steam injector are a very short heating up time, the ability to handle media with suspended solids, low capital cost, easy cleaning and maintenance, and high steam utilization efficiency. The disadvantages include the potential for foaming during heating and the fact that direct steam contact requires allowances for condensate dilution and necessitates the use of very clean steam, free from anti-corrosion additives. Decision flow chart for processing a contaminated fermentation. Finally, we have a decision flow chart for processing a contaminated fermentation. This chart guides the decision on whether to continue with a batch that has been contaminated. It starts with the question, is the level of contamination so high that further processing is unacceptable on quality or economic grounds? If yes, the batch is rejected. If no, the next question is the contaminant a potential pathogen? If yes, reject. If no, will the contaminant survive the downstream processing? If yes, reject. If no, is the contaminant known to produce an antibiotic or other hazardous metabolites? If yes, reject. If no, is the contaminant known to produce a toxin or toxic substance? If yes, reject. If no, does the contaminant pose a quality risk in the final product? If yes, reject. If no, the flowchart continues with similar risk assessment questions, each leading to rejection if the answer is yes, until a final decision is made to accept or reject the batch based on a comprehensive safety and quality evaluation. Thank you for following along with this comprehensive session on industrial sterilization. We've covered its critical importance, the various methods available, and the detailed design considerations for both batch and continuous processes. Mastering these principles is key to successful and contamination-free fermentation. Thank you. See you in the next class.